3D genome organization was shown to be intimately linked to its biological function. Changes in nuclear architecture can affect cell fate, and disruption of genome topology can result in pathological phenotypes. Regulatory interactions between specific regions determine whether genes will be activated or repressed, and thus are crucial to establish and maintain cell identity during development. However, these interactions are difficult to study genome-wide because of the high complexity of possible contacts inside the nucleus, where more than 2 meters of DNA are packed inside a tiny sphere of about 10 micrometer diameter. In this study, we focused on neural differentiation and cortical development as a paradigm and performed ultra-deep coverage high-C on mouse embryonic stem cells, neural progenitors, and cortical neurons. Furthermore, using transgenic mouse lines, we purified neuroprogenitors and cortical neurons directly from the developing mouse embryonic neocortex in vivo. We found that gene activation is correlated with chromatin insulation and long-range interactions, but artificially turning genes on is insufficient for creating new domain boundaries. Furthermore, highly transcribed exon-rich genes frequently engage in long-range contacts both in cis and in trans. In addition to these findings, which were valid for both pluripotent and differentiated cells, we observed cell-type specific regulatory interactions. During neural differentiation, contacts between active topologically associating domains become less pronounced, while inactive topologically associating domains interact more strongly. An extensive polycomb network in stem cells is disrupted during neural differentiation, while dynamic interactions between neural transcription factor binding sites are established. Finally, cell-type specific enhancer-promoter contacts are established concomitant to gene activation. These findings illustrate how 3D nuclear architecture is deeply linked to the normal physiological and pathological function of the brain in vivo. They suggest that 3D chromatin architecture represents an important layer in the regulation of gene expression and have implications for several brain-related diseases such as intellectual disability and autism, which are frequently associated with chromatin remodeling complexes.